Hello, I want to welcome everybody to today's lecture by Dr. Shirley Pomponi. I'm Dennis Hanisak, a research professor here at Harbor Branch and host of the series. In 2 weeks, on Wednesday, March 10th, our lecture will be Ocean Color Remote Sensing, Observing a Changing Planet, and will be presented by Dr. Tim Moore. Tim is a research professor here at Harbor Branch. He specializes in using ocean color remote sensing technology to study the ecology of phytoplankton in the environment. To do this, Tim works with images taken by satellites. You will learn from his talk how this technology works and how it can be used, with examples being the harmful algal blooms that so many of us know about in the northern IRL, Indian River Lagoon, and Lake Okeechobee. I've seen these images. They are dynamite, and if you see the images, you'll have a much greater understanding of what's really going on with those blooms. So I encourage you to come and hear Tim Moore. Today's lecture is on a project that has been quite a team effort. The talk itself will be given by Dr. Shirley Pomponi, the lead principal investigator on the project. For the questions and answer period, she will be joined by the rest of the PIs of the team, John Reed, Amy Wright, and myself. Shirley is an ocean explorer and a marine biotechnologist. She was our executive director at Harbor Branch from 2004 to 2009, and then director of our NOAA Cooperative Institute for Ocean Exploration, Research, and Technology until 2018. John has led research and conservation programs on deep water and mesophotic coral reefs for his whole career. And indeed, that's been quite a career. He has, in fact, worked at Harbor Branch longer than anyone else ever. John's research and outreach efforts resulted in the establishment of marine protected areas for both the Oculina and Lophelia coral reefs. Amy has directed our natural products chemistry group for many years. Amy is a wonderful chemist, and she holds many patents on the compounds she has discovered from the sea. The primary focus of her research is the discovery of natural products useful in the development of novel therapeutic agents for the treatment of cancer and infectious diseases. I'm the plant person on the team, and most of you know I'm focused on macroalgae or seaweeds and seagrasses. And I will just say I have had many wonderful opportunities and experience thanks to my 3 colleagues. So here's a fun fact. I thought about this the other day. The 4 of us collectively have worked at Harbor Branch for 165 years. Sounds like a life sentence, right? 165 years. That's really 4 lives. And of course, we're still counting. So today, all of us are connecting from different locations for our lecture on the Marine Biotechnology Reference Collection, a new web-based resource for research. Here's Shirley.
Thanks, Dennis. Everyone can hear me okay? I hope so.
That's a yes.
Okay. All right, let me just share my screen. First of all, thanks very much, Dennis, for that very nice introduction. And thanks everyone for joining the lecture today. And this has been a fun one for us to put together. I'm kind of the spokesperson for the group. Hopefully we'll all be able to get together as a panel maybe later on in the year and talk about some of the fun things that we've done collectively as well. Okay, so I'm going to share my screen now and go through and hopefully, let's see, there we go. Okay, so again, Dennis gave the introduction. We have this incredible collection that was made since 1984. And we have, an incredible number of samples in that collection. And I'm going to give you a little bit of the background of it. I think many of you already know about that, about the Marine Biotechnology program, because you've heard, in fact, just even recently, Esther Guzman talked about some of the drug discovery research that she's doing as well. So let's see, why is this not working? Well, let's see, there we go. Okay, so since 1984, we've been able to collect about 30,000 samples of benthic invertebrates, so algae, sponges, octocorals, echinoderms, you name it. And I'll show you some photos of all of these later on, primarily for our drug discovery research program. So it ended up being a really unique collection, because when we collected these samples for the drug discovery program, we wanted to make sure we had a reference sample, so that if Amy and her group found something interesting with biomedical applications, we knew from what sample that came. And so we start building up this taxonomic reference collection, which is really diverse. We have algae and invertebrates, 13 phyla. We have individuals of the same species that have been collected from either the same location or different locations and depths over the last 30 plus years, actually. And more than a third of those are from deep water, so collected using the Johnson Sea Link submersibles. And we're going to do a little dive here. I have a video. I get really scared when I have to embed a video in a presentation, but we practiced it numerous times, so hopefully it works. So we're going to do a little video of the dive. And most of the dive is actually being narrated by Amy Wright. So let's see how this works now. This is when we had two of the three that we had. Very rare to see both of them out at one time. Probably just a photo op. But you can see on the front of the ship the Johnson Sea Link submersible. And just in a few seconds, you'll see the submersible being launched. There it is, getting ready to go in the water. I think many of you.
Roger, we have a seal.
But this is sample number 2. You can, we're collecting one up at the top, so that's at the top of the screen, and the one that's the center bottom that's brownish and spherical is the same thing. Very, very pretty. Sample number 7, 2,984 feet, here in just like a fabulous place. And the other one came so easily. She's growing right on this vertical drop-off. Should get that on there too. Oh, collection of sample number 1. We're working right on this vertical edge. It's just the drop below us, it's just black if I look under my feet. There's just nothing. Oh no, don't drop. Catch it, catch it. Oh, Dan has a career in baseball. Just kind of an upside down something or other. And looks like a lithistid, but I'm not really sure. It's probably going to be brittle. It's just so big, but maybe just get part of it. I just want to get out and scrape it off the rock. Oh, man. Look at that. I think that might be Spirostrella.
Might be what?
That sponge Spirostrella. I don't think I've ever seen it this deep. 12. Good job.
Yes, he's jumping off the cliff here, look. I've got you loud and clear, huh, me?
Loud and clear, 340.
Okay, so that was just a real quick dive. Usually our dives lasted about 3.5 hours. And then we would come back up on this ship and the batteries would get charged And that during that period of, and then we would go out again. And then during that period of time, we did what we called processing of samples. And so there was a lot of work that was done. Here you can see in the upper left, John Reed carrying one of the buckets from the sub that had samples in it. And on the lower right, Jennifer Sandel also. And then we would take them back into the wet lab and start sorting them. And you can see in the upper left, I think probably Amy was on that dive. And so she's trying to match up the sample numbers that you recorded in the notebook with what's in the bins, what's in the buckets. And then we would assign numbers to everything, print out labels, and then start making subsamples, taking samples of each of the specimens. And in fact, in the lower middle, the bottom middle, you can see like test tubes and stuff there. And you can see a lot of people in the background because there was just a flurry of activity. People are taking samples for chemistry. In fact, let me think, I have that here. Yeah, we'd have taxonomic vouchers, so reference vouchers. And you can see in the upper right a photograph of one of the rooms that we have these samples housed in. So we have taxonomic vouchers that are in ethanol. We have samples that were frozen for chemistry. We have ethanol extracts that you can see in the picture on the lower left. And then these were used for bioassays in the drug discovery program. And then we also had samples, subsamples of each one of these specimens that were cryopreserved, frozen for microbial isolations. And then anything that was left was put in the freezer for subsequent work on the chemistry. So if we found something really interesting, that we had enough material to go back and extract some more of it. So, you know, it ended up being a really, really interesting collection and really unique. So we had about 59 or 60 expeditions over the years, about 1,400 submersible dives. And most of those submersible dives, we have videos of the specimens that we collected. So it's pretty cool. So in addition to photographs, we have videos, and then we took videos. We took photographs of the samples like back on the deck in the wet lab. All the videos now have been annotated, and I'll talk a little bit more about that in a few minutes. And we have about 30,000 images. So we started talking about this, and you know, Dennis. I'm not going to put Amy in this category, but Dennis, John, and I are not. We're late career scientists, so. Everyone recognizes the value of this collection. We all recognize the value. We have colleagues who've asked to borrow samples from the collection because it's such a unique resource. But every time someone asks for samples, we had, you know, go into our database, print out records of what we had, send them the samples to study, and so on. And we decided to submit a proposal to the National Science Foundation, and their Division of Biological Infrastructure provides support to researchers who have collections like ours to digitize. So in other words, take those samples, those like the images and the videos, and digitize them and make them publicly available on the web, make them web accessible. So we thought this would be a good idea, and it has turned out to be a really great idea. So I'm going to give you some insights about what it is that we're doing, what the website is all about, where we've been, what we've collected, and why it means so much to us, and why it's an important resource as well. So this is a map that's been generated by the website. And I'm going to go through and show you some details of it in a few minutes, but every place you see a yellow dot are places where we've gone collecting. I mean, literally all over the world. Some of these have just been what we call land-based collections. So we fly somewhere, and then we either charter a boat or, you know, go out and go diving in a place. So shallow water collections. But a lot of them are using the Johnson Sea Link submersible or remotely operated vehicles. And so if I expand that, if I go in a little bit closer, you can see a lot of dots around Florida, the Bahamas, the Caribbean. And if I go in and even closer, and these are some of the functionalities of the database. So you can actually go in closer and closer, you can actually click on a dot and see what we have from that site. So here we're going to Roatan, which is off of Honduras. And now I'm going to take you, I think I'm going to take you into the database. So these are areas off of Belize and off of Roatan, Honduras, where we've done collections. So again, this is just another map of the sites where we did collections. And now I'm going to actually introduce you to our data portal. And in the data portal, what I've done here is I've selected, and I'll take you through this in a minute or two. I've selected on the left-hand side, the country, Honduras, the depth zone. I wanted to see things that were in deep water. I'm a sponge person, so I selected Porifera for the phylum, and that's all I did. And then I went across, if you look across the top of the slide, you see a bunch of different tabs. And I clicked on the tab that says In Situ Image Gallery. And then that In Situ Image Gallery, then when we call that up, it shows me, and we can scroll through it. I'm not going to now, but you can scroll through it and you'd see all the different sponges that were collected from deep water in Honduras, in Roatan. This is very useful. So, because sometimes, for example, I might be interested in just like a glass sponges that were collected say in the Bahamas, and I'm not a glass sponge expert, so they haven't been identified in our database, but we have other experts who are interested in this, and so they can look through and say, oh yeah, I want that. Well, this is just, it's a bad example, but I want that yellow sponge that's in the upper row, and then start going through the pictures and selecting and saying, okay, this looks like what I'm interested in. This looks like what I'm interested in. So here's the yellow sponge. You can actually click on that image and you can get the actual in situ or underwater image. And this is just a montage of photos of sponges, just to give you an idea of the diversity that we see. So there's a diversity of color, of shape, of size, really incredible. And you've heard me, some of you have heard me talk about sponges before, but there are nearly 10,000 described species of sponges. And we have a lot in the Caribbean, the tropical Western Atlantic. So I'm just going to show you some different things that we've collected. And I'm going to show you in situ images. So the underwater images that were taken either by the Johnson sealing or by ROV. So this is a sponge. Actually, this is a sponge called Discodermia, from which a bioactive compound discodermalide was isolated. This is a long tube sponge, Naplicina. This sponge is another one. It's called Spongoceroides. It's one that a really interesting compound called the topsentins that have anti-inflammatory and antiviral activity comes from. Okay, this one might look like it's a sea fan, but it's not. It's a sponge. It's one of the prettiest sponges, one of my favorite photos of a deep water sponge. But here's an octocoral, here's a gorgonian, and here are some more. This is a crinoid. Here's some more I'm going to show you a few echinoderm pictures now. We frequently see these really interesting associations, like echinoderms on sponges, on octocorals, lots of different associations here. This is a deep water glass sponge, and with brittle stars around it. This is an anemone called the Venus flytrap anemone. Really interesting sea star. We're really fortunate because when you're in the sub, you can observe these interesting behaviors as well. So, if we had just collected this sea star, it would be flat. We wouldn't really have had the opportunity to see it, kind of almost like walking around. There's a cluster of urchins. This, I think, is a screen grab from a video. And this is a sea cucumber, actually, a swimming sea cucumber. Interesting associations here. You can see shark, decapod crustacean, fish. This is a really interesting shell. It's called a slit shell, which actually is very collectible. There are shell collectors who value highly these types of shells. And it's on and actually eating a sponge that has lots of silicious spicules. And I can't imagine why something would want to eat something like that. It would be kind of like eating fiberglass. Here's a little crab on a octocoral, beautiful albino lobster. And I just want to remind everyone that although I just showed you a bunch of photos of deep water organisms, we also did many, many collections using scuba in shallow water. So, you know, in caves and crevices and along walls. One of the nice things about the submersible, the Johnson Sea Link, is that we were able to operate in areas where you wouldn't be able to normally collect things with like a trawl or a dredge. So on these steep slopes and stuff. And we also like to go, when we went diving, in some unique habitats. So walls and crevices and caves and so on. And here are some photos of shallow water sponges. This is a shallow water sponge, more sponges. You can tell that I'm very prejudiced here about sponges. There's an octocoral. Okay, I'm gonna just take like a few seconds, and because the next part of this presentation is gonna be about the actual website. For those of you who can multitask and bring up another window on your computer, if you want to play along and try out this app, you can. And don't worry, it's going to show up. This hyperlink will show up, or this URL will show up in the next few slides. So while you may be checking that out, trying to load up the application, I wanted to take the opportunity to acknowledge my colleagues. Dennis, of course, introduced all of us, but I will say that this project has truly been a group project. Dennis and Amy and John have really worked very, very hard on multiple aspects of this project. John has, I think probably John has kind of provided oversight for just about everything on the project, the database, the photos. He, I was going to say single-handedly, but almost single-handedly, actually edited and cleaned up about 50,000 images, I think. Amy is close second on that. I think she probably did about 20 or 30,000. Dennis has been our website guru, really providing oversight and direction and design of this website that I'm going to introduce you to right now. We've all worked together for many years. I consider each of these folks not only close colleagues, but close friends as well. Okay, I want to also acknowledge many other people who, without whom this would not have ever happened. Jim Fredlock and Barbara Mills volunteered for about 15 years. They started in about 2004, and between the two of them, digitized 120,000 photos that we had taken. So back then, when we first started in the like 1984, it was all slides. We had Polaroids and so on. It's only been more recently that we had digital cameras, and so the images were automatically digitized. But Jim and Barbara were, I mean, just the dynamic duo in terms of getting us to the point where we have, I mean, literally over 100,000 images that they digitized. So Jim passed away last year. Barb, I think Barb is watching, so thanks very much, Barb. Stephanie Farrington's our database guru. She's the one who really cleaned up our database so that when we handed over the data to Brian Botson, it was cleaned up. Everything was kind of consistent. The taxonomic names coincided with what's the accepted taxonomic name. Brian and his team, Brian Cousin, Jennifer Sandel, Elizabeth Irvin-Gadamke, were responsible for digitizing and annotating the videos. This was a huge, huge job because we were taking all of these videos. You heard Amy narrating, and so, but we never kind of transcribed those notes into a digital format. And so they went through and painstakingly watched every single one of our videos, hundreds and hundreds of hours of them, and annotated what anybody who was, who was narrating the videos. So sometimes it was me, sometimes it was Amy, sometimes it was John. There were others in the sub. And they actually annotated all of these videos so that now we can go back and click on, let's just say we want to look up find a sea urchin, and we can find that based on what they've done in terms of annotating the videos. So thanks very much. And Brian also made smaller video clips of all of our videos, so that we can upload small clips on our website. Again, I can't overemphasize how much work this involved. And so I want to thank each one of them. And then finally, last but not least, Brian Botson's the one who took all of these data, the digital data, the images, the videos, and created the website that we're going to go through right now. So again, thanks very much. Okay, so this is what it looks like when you go into the data portal. If those of you who are on the website now, you can go on that data portal. And you'll see over on the left-hand side, there are a number of, I don't know if you can see my arrow or not, but on the left-hand side, there are drop-down menus. So you can select a country, for example. So let's just say we select Honduras. And so you click on that, and then you can select the depth zone. So you can select whether it's shallow water, mid-level, deep water. And in this one, I already selected deep aphoda because I wanted to see deep water. I wanted to see deep water sponges. And in the in the phylum box, there's another drop-down menu, and you can select one or many phyla. I selected peripheral here. So then you can see, let me see if I can do this. No, let me just go back. So then what you can do once you're in that is, let's go across the top of this, of the portal. So this is our taxonomy data table. There's another table that just shows information about the location, latitude, longitude, and so on. All data is another tab that has all of the data. There's a tab where you can select on deck images, and so you can see what the samples looked like when we brought them back up to the wet lab, and an in situ image gallery, which I already showed you. If you click on that in situ image gallery, it shows you all like a montage of all of the images, and then the map. And the map is, and in a little while, if we have time, we'll actually do this live. I didn't want to risk doing it by screen sharing, but we'll try and do that after the presentation. Okay, so let's see. How are we going to use this resource? Why did we even do this? Well, first of all, as I mentioned before, some of our colleagues, especially people working in sponges and octocorals, black corals, trying to think what else. Even sometimes we had like little bycatch where we had little cup corals, knew that we had been doing this work in deep water and had been to areas where no one had ever been. And by the way, I want to point out that all of these collections were made with permits. We had to seek permission, request permission to do the collection. So all of the collections that we made were permitted collections, and that's really important now to be able to document that as well. Anyway, so my colleagues in the sponge world would say, okay, I'm really interested in all of the, let's just say rock sponges that you collected. And so we would go through, John would help, we would go through, Amy would help go through and select kind of like a subset of these, send them photos, they would select some samples and that they were interested in. And then they'd either come to Harbor Branch and work on them, or we would ship them samples to study. But and more, our colleagues, our collaborators and colleagues we're starting to ask even one, really recognizing how important and interesting this collection is. And so we were getting more and more requests, and mostly requests for information as well. Did you ever collect species A from the Gulf of Mexico? And if so, at what depth? And we could find that, John, Amy, Dennis, and I could find that ourselves. But we really wanted to be able to make the website of the data available to a broader audience. And let's face it, we're not going to be working forever. John and Amy and Dennis and I, well, maybe Dennis and Amy, but I don't think John and I are going to be working forever. So how do we use this resource? So we recently completed, I was on a National Academy study where we looked at biological collections and what they've been used for, how they can be used in the future. And they looked at collections of all kinds. And our collection was one that, not ours specifically, but the type of collection that we have, which is actually like considered to be a private collection, because it's not in an archival institution. It's not in a museum. It's in our museum, but not publicly available. It was project-based. So the collection was made for a completely different purpose. It was made as for taxonomic references for a drug discovery program. And so, these are what we're doing with this now is kind of an unanticipated use and a benefit of having this really unique collection. So it gives us the opportunity, for example, since we've been to, some places multiple times over the years, the ability to look at organisms, say, for example, through time and say, okay, can we get some idea about impacts of climate change, for example? Resource managers can go to the site and say, okay, I'd like to see everything that's at this particular protected area in the Gulf of Mexico. Really useful tool for education and public outreach as well. And I just want to point out one thing. It's kind of a biased collection. So we really were interested. I mean, we didn't collect stony corals. We didn't collect anything that we were not allowed to collect. And we were really focused on larger things, so not little tiny crusts. So it's kind of biased in terms of looking at benthic invertebrates and algae that are a little bit larger in size, so that if we found something that was interesting, that we would be able to extract the compounds from it. Many of the images that you'll see, if you look at the images on the site and actually even look at some of the videos, there are many interesting associations that have not been studied. And this, I'm showing you this picture. This white thing on the top of that kind of pinnacle is a sea cucumber. And we ended up going back and collecting, searching for the sea cucumber. When we first collected it, it was kind of bycatch. I don't know if it was I or John, but one of us had been collecting a sponge, and kind of a bycatch from it was a little tiny sea cucumber. And so it wasn't an intentional collection. And Amy and her group tested it and found that it had some really interesting compounds with biomedical potential. So we were able to then we have that search image on what it is that we want, we were looking for, and we're able to actually go back and look for this organism, because we just figured, and Amy may comment on this later, we just figured when we saw that, it's like, oh, well, this came up, it was in the sediment somewhere. We never realized that it was actually living on either corals or on sponges, and not actually burrowing or moving across the bottom. And a really, I think, kind of a side benefit of the videos that I had not appreciated really until Amy really was the one who pushed this a lot and said, just looking at those videos, there's a lot of other stuff in them that we did not collect. So when I mentioned to you that we had a very focused collection, there are a lot of things we didn't collect. And there are people who might be interested in, in echinoderms that we didn't collect, or in crustaceans that we didn't collect, or mollusks. And you can see those in the videos as well. And the fact that they've been annotated now, the videos have been annotated, makes it a lot easier for us to search on those videos as well. Stephanie Farrington gave us this information yesterday. It just made me feel like, oh my gosh, I have a lot of work left to do. Most of the samples in our collection have not been identified to species. I was actually kind of impressed because 34% of them have been identified to species. So that's way better than I thought. And if you look at genus and species between the two, we have about 65% of the samples that have been identified either to genus or genus and species, but there's still a lot of work to be done. And what makes this collection very unique and this website unique is it's not just a website with photographs and videos and data. It's a specimen-based website. It's a specimen-based collection. So not just collections of photos and videos and data. So if someone is interested in studying one of these specimens, we've got it to study. And that's what makes this, I think, really, really interesting. So I want to, I'm not through yet because I'm going to show you some more pictures after this, but I wanted to stop now to acknowledge the army of people who contributed to this, to this product, really. I mentioned that video digitization and annotation was a huge job. Brian, Jennifer, Elizabeth, Kate, I didn't have a picture of Kate, but Kate Beckett also helped. And image digitization, selection of those images and editing was done by Jim and Barbara. I mentioned the two of them. And then of course, John and Amy. Video clips, Brian, you're a hero, really. The web database, Brian Botson and Dennis Hanisak, and then we have a Microsoft Access database, and I have my name there, but I'm not really the guru. The three gurus of that Access database are John, Stephanie, and Amy. But all of our data are digitized in that database. So, and then another aspect of this project was to go back into our collection, the museum, and barcode all of the jars, all of the specimens. 33,000 of them. Okay, so this has been a huge job. So Brienne Varnerin, Megan Conkling, Jennifer Sandel, and Cassidy Dugan. In fact, Jennifer and Cassidy are still working on doing the barcoding of those so we can keep accurate track of where everything is. Of course, there are many colleagues, staff, students who have participated in our expedition since 1984. Some still here, some have passed on or moved on. But it's just been absolutely an incredible journey. And of course, none of this could have happened without having the ship and submersible crews and the remotely operated vehicle crews. And I just want to make one final acknowledgement, and Amy actually brought this to our attention a couple weeks ago. Back in 1984, maybe even a little bit before that, Seward Johnson Jr., who was the chairman of the board of the Harbor Branch Foundation, had a vision. And his vision was to establish a marine natural products drug discovery program. And so Sea Farm was born then. That's when it came about in March of 1984. Amy and I joined the group. Amy joined in September of '84. I joined in December of '84. So we've been around for a while, but it was because of Mr. Johnson's vision and his support, financial support, that we've been able to make these collections. Most of these collections have been made and have been supported by the Harbor Branch Foundation. So, there were years when we went out to sea like 80 or, 100 days a year. And let me just back up a minute, because our sponsors of this were NSF sponsoring this project to make the data publicly available on the website. Cruises have been supported by NOAA, the Ocean Exploration and Research program there, and through our Cooperative Institute, also funded through Ocean Exploration and Research. But Harbor Branch Oceanographic and the Harbor Branch Foundation have really provided a lot of additional support for us to do the other work on this. And I might want to also mention too that the foundation has provided some support for digitization of videotapes from like the Oculina reefs, areas off the coast of Florida. And John's been really instrumental in making sure that those really old videotapes. I mean, 40-year-old videotapes are preserved, and have, with support from the foundation, we've been able to do that. But again, I wanted to make sure that that and we express our appreciation. And Mr. Johnson passed last year, so I think he'd be really pleased to know that not only have there been some really incredible discoveries from the drug discovery program, but that there have been these side benefits as a result of the collection as well. So I want to just take a little bit of time now to just go through a few images. If I have some time, few images, just to show you some of the people who've been involved over the years, been involved in going out to sea. We've had a lot of fun. One of the best, I think is that Dennis? I think that might be Dennis, and the Galapagos, and there's Klaus. One of the I think the best things about doing this project, and I imagine that Dennis and Amy and John will agree as well, has been to be able to go back and look at these images and really, first of all, think, oh my gosh, how lucky have we been to be able to go to all these places, with people with whom we enjoy, working, and just having a lot of fun. It was a lot of work. Our days at sea are long, long days, 18-hour days, and we're working all the time. But we enjoyed what we were doing. We were having fun doing all of this work. This is a baby doll. There's a story behind this. Maybe John will talk about the baby doll parts, but John had this kind of collection going of baby doll parts that he's collected from pretty much all over the world. Here we are. I think this was in Galapagos. Our reconnaissance trips when we would go out scuba diving were epic. We couldn't get in the water until John had selected just the right place, and it usually involved John jumping off of the dive boat and being pulled around, and just looking over the bottom, and then signaling when he wanted the boat driver to stop. This is Ed Armstrong, who passed away several years ago. It was because of Ed that I'm actually working at Harbor Branch. Ed's the one who actually recruited me to come down and be part of the Sea Farm project. Again, here's John. It was a lot of fun when the sub crew would let us participate in a recovery. Jill, we like to give our staff and students, this is Esther, she gave a talk a couple of weeks ago, the opportunity to be able to get in the sub as well and do the collections. Over the years, we've had several students and interns who've been able, and postdoctoral fellows who've participated. There's Tara, Alaire, John in the sub. Usually see us laughing. You don't usually see us frowning, even though it was a lot of work. We really enjoyed our time out at sea. I think that's it. That's it. So thanks very much for your attention. I hope you enjoyed, first of all, thanks for allowing me to reminisce over this, and I hope you enjoyed this. And I'd like to now open it up, bring Amy and Dennis and John on, and I think we're going to have Q&A now.
Well, thank you, Shirley. I'll clap for everybody out there.
Clap for Shirley.
And the guidance for the audience is the same as it always is, but if you're new, you can type your questions in at the bottom. Just go to the bottom of your screen. You can find something called Q&A, and you can type your question up. And then I go through them, and I'll go ahead and throw them out in most cases to Shirley initially. If anybody has a question that you'd rather address to somebody else, go for it. Otherwise, Shirley will kind of guide us to them.
Okay.
So the first one is from
Go ahead.
one of our graduate students from Greer. two really good questions. First one is, do you have the name of that sea fan looking sponge you mentioned from the in situ photos?
Yes, it's called Placalia, Phakellia.
And
It's an
then the one I'm really curious to hear the answer to is, we know you're an avid diver. Do you prefer diving, like scuba diving, or would you rather do a submersible dive?
So that's like asking if I would rather have chocolate ice cream or vanilla ice cream. I actually do have a preference over that. But anyway, I enjoy, I think for me, what kept me at Harbor Branch all these years, 36 plus years, was not only the amazing people I worked with and the things that we did together, but the fact that we were able to go to some incredible places using the submersible. So to me, that's been a game changer. I think we've all been incredibly lucky to be able to have that resource.
So this is a question from Madeline, and she wants each of us to answer. Let's go with Amy first, okay? But we'll take turns on who goes first. These are all going to be hard questions now. What was one of your favorite collection locations? I'm sure there are many, but
so there are many. I don't even think I can just say one. And then when we got to get all the videos digitized recently, I started watching all these old videos, and I'd spend 45 minutes of a dive just watching, and they were all amazing and fabulous, as I said in that video that was there. Anywhere in the Bahamas was always great. Down in the Portales Terrace, which is at the south part off the Keys in Florida, is absolutely amazing on these kind of sea mounts that only come to 500 feet or so from the surface. And yeah, there are just too many, but anywhere in the Bahamas is amazing.
the Bahamas really is better, isn't it?
Yeah, it is better.
So John, I gave you some extra time to think.
Uh-huh.
What was one of your favorite collection locations?
Same as Shirley and Amy and all of you guys, every location was different, and everyone was spectacular. To know that we, by and large, we were the first people to ever be there, ever to see it live. It was amazing. Bahamas, yes. The outer islands of the Bahamas, San Salvador, which is the far eastern part. There's my cat. Far eastern part of the Bahamas is spectacular. Roatan, the Galapagos. I think one of the coolest dives is when we inadvertently landed or went into a volcano off the Antilles, Kick-and-Jenny volcano. That was a kick, because I got blown out the top of the volcano with a hot air or hot water plume coming out of it. So that was fun.
Tell the truth, John, though, when you heard it was called Kicking Jenny, didn't you actually want to test the hypothesis? You really went in there wanting to know if it had any kick to it, and you found
We
out.
didn't know the name of it then at that point.
Oh,
We just saw a ridge on the fathometer and dove on it, and then realized, hey, this is circular on top. Why is it circular and have a hole in the middle? And it's a volcano.
so for me, I would also favor the Bahamas, specifically San Salvador Island. I'm a plant guy. That's the place in the entire world where algae are attached and photosynthetic deeper than anywhere in the world. So that's 286 meters, which is 860 some feet. But my first dive was at Pulley Ridge. This is going to kill Stephanie Farrington, who's on watching this, because she hates it because it's so flat and so boring, she says. That's because it's totally covered by algae. And it's a long story as to why I went and how I found it, but that was the place I've gone back to over the years, over and over again, just because I think it's spectacular. Shirley.
So it sounds to me like we need to organize a trip to the Bahamas pretty darn soon.
I
So
suggested that.
I know. Because for me, I mean, no question in my mind, it's San Salvador is my absolute favorite place to scuba dive and for the submersible dive. The best submersible dive I ever had was there. You know, the sub was just trimmed out just perfectly. There were all these buttresses and canyons and stuff. It was just fantastic. And then scuba diving, you can go down and I like kind of swimming through these like little crevices, and you can go, I think maybe it's about 40 feet, John. I don't know, 40 feet. And then you can swim through this little kind of tube and you end up at about 100, you come out at about 100 feet. I love that kind of diving. So yeah, absolutely amazing.
So the second question, and we'll go in reverse order, so Shirley gets it first this time.
Okay.
What would be a location you haven't visited that you would want to collect from?
Well,
where would you want to go?
okay.
You haven't been
So I'm going to tell you, because this was always on my bucket list, but now that I've gotten older and I don't like being in cold water, but what would have been a place where I would have loved to have gone would have been the Antarctic and diving under the ice.
Okay. For me, hard to say, but we were supposed to do some deeper dives in Brazil at one time near the end of the JSL time. And there was one place that we really wanted to go to that we never got to. And the other cruise that we wanted to do, I think most of you remember, around the turn of the century, the turn of the millennium, we planned to take the ship and the submersible to Australia and New Zealand. And that would have been like a very long cruise, and it was going to be very expensive, but we almost figured out how to do it.
Yeah.
I think that would have been a pretty amazing thing to have done, those two locations. John, what about you?
Well, you know, we were always kind of limited how far we could go on the ship, in a certain amount of time. But we were able to go to the Pacific Ocean twice, to go to Panama and the Galapagos, which was, truly awesome. Then we went to some other sites in the Pacific on other ships. We were on a Russian ship to American Samoa and also to the Seychelles. So I think I would want to go back to the Pacific Ocean. Certainly Australia, New Zealand would be awesome. But Hawaiian Islands, that whole chain of Hawaiian Islands would be awesome.
Yeah, indeed. And there's some good, there's some good submersible, but shallower submersible work going on out there. Okay. Somebody has asked, how many sponges weren't identified? So Shirley, I think that's for you.
Well, except Stephanie just put that together. John, do you have that information?
Yeah.
Okay. The short answer is too many. In fact, actually, I'm going to just jump in here. Dennis looked at this list yesterday that I put up, and he sent out an email to John and me, and he said that if John and I each identified 10 specimens per day.
Only 10, yes.
Only 10 per day each, that we would be done by, I think, May of 2023.
Yes, it's going to take 2.5 years.
Right.
So.
Okay, go ahead, John.
Well, I mean, a breakdown of the 33,000 samples that we've collected. We have 3,400 algae that Dennis needs to identify. We have 200 arthropods, which are crustaceans like crabs and shrimp, 1,000 tunicates, 6,000 cnidaria, which are like coral and sea fans and black coral, 1,400 echinoderms, 500 mollusks, and a whopping 18,000 sponges, of which we have about, 8,000 to identify the species. So we've identified 11,000 of the 18,000 to species level. So figure it out.
Okay, this I think is probably maybe for Amy. The question is, what medical products have actually been derived from Harbor Branch?
Okay, so so the one that I think we really claim that we had at least some role in, although we didn't do anything with the clinical development part, is a compound that's now marketed as Yondelis or Tribectidin. And it comes from a tunicate that you can even find on the docks here in Fort Pierce, but it's much more common in the Florida Keys. And in fact, on my very first expedition ever, when I just learned to scuba dive, I went with Shirley into mangrove islands in the Keys, and we collected that sample and we put it into an animal model for cancer and found 9 out of 10 cures in that animal model. So we started doing work, and our contribution really was to figure out parts of the structure that had been stumping people for about, gosh, I don't know, like since 1967. People had been trying to do the structure, and we completed a major portion in 1990, and there were other people. Ken Reinhardt was part of Sea Farm, who was working on it as well. And it was then, Ken actually had a patent on it from his University of Illinois, and it went forward with Pharmamar, a company in Spain, developed it, and it is now approved for soft tissue sarcoma in about 78 countries. And what's even more interesting is that they've now done a synthetic modification of that structure, and they have that it's really outstanding for lung cancer. And it was just approved last year for lung cancer. So this new, and it's Tribectidin. So I think those are the ones we claim. We've had others go to clinical trial. Sarath Gunasekera found a compound, discodermalide, and this team did a lot of work on discodermalide, collecting it, and Sarath was the chemist behind that. And it went to the clinic, but didn't make it the whole way. And then we always are ever hopeful that something, the next one that we have, will make it. So we have quite a few interesting things right now.
Well, thanks.
Long story.
Yeah, and I guess you have the freezer full of samples, so you
Yes.
can go on and on for some time, I guess, to continue to look for new opportunities as new technologies and new ways of doing assays, I guess, evolve, right?
Yeah, as long as we keep getting grants.
I think if there was ever a motto for our collection efforts, it was we would be out there. I don't know how many times one of us or somebody else said it, but we would look at something and we would, even if we were like tired, like Shirley said, after those long days, we would just say, it could be the cure. I mean, that was the whole idea. It really was exploration.
So Amy, I have a question for Amy. I mean, I think a big part of the BMR collection is your library of like individual that you're able to send out little tiny pieces, or you can tell about it to different people, different different organizations and universities that are doing biomedical research. And you can send a whole library of samples to these people to run through their assays to look for different, not just for discovery of the cure for cancer or something, but discovering biomedical pathways, how these things work. How does this chemical work to kill or stop a cancer cell? And I don't know, maybe you can just talk about that a little bit.
Boy, put me on the spot. Yeah, so, well, we have something that's called a enriched fraction library. So we took a lot of the organisms and we made them less complicated mixtures than if you just make an extract. That's like making a cup of coffee and there's all kinds of stuff in there. So we make it less complicated than that. So just fewer chemicals per vial. And we send that to people all over the place. So we have, right now, we've been looking at cryptosporidium with Robbie O'Connor, who's out in University of Washington. And that's a disease that kills people with AIDS. It's one of the infections they get that there's really no treatment right now. And we actually have just found 2 or 3 compounds that look really good in her assays. And so we just keep doing that and sending them to different people. People look at how they're, what the mode of action, how they kill cancer cells, or we have a program for antibiotics that we work on with Rutgers for these new and emerging things. We sent some of our materials out for COVID testing, and we got 2 really nice hits. They block infection, but they don't work if you're already infected. So we have to figure out what to do with that. But yeah, there's just so many opportunities, and there's a lifetime more for many other people besides just me.
Yeah. Okay. So this is a question came in. Seeing the data portal reminded of the EPA database Ecotox. So I don't know if any of you are familiar with that or not. And I was thinking that the addition of bioassay info could be really interesting for a data scientist project, looking for patterns related to taxonomy and bioactivity against different targets. So I don't know if you want to address that, Amy or Shirley. I think one of the 2 of you might be best.
You want me to take it? I'll take
Sure.
Sure.
Okay, so right now, and Marissa, thanks for that question. Right now, we don't anticipate putting those data up. And I'll tell you, really, the reason is that we're working on a lot of those samples now, and we invite collaborations. So if Amy's group collaborates with a number of other scientists worldwide to use the samples and the data for looking for other for activity against other disease targets. But right now, we don't have plans to do that. I understand what you're saying, and yes, it would be, but I will tell you that we have searched through. One of the things that Amy and I and John have done over the years is search to see if there are some patterns for bioactivity. Can we see, if this one particular type of octocoral produces this really interesting active compound, do related species do that, or do all of them do that? And we haven't so far. Within a species, of course, you can see that, but typically we haven't found patterns of different types of bioactivity as well. Amy, did you want to add to that?
Well, so part of it would be that, so we got this one grant from NSF to pull all this data together and to get that all totally organized. And we would need funding to do that because it's a huge job. We have probably put things through, oh gosh, I don't know, 40 assays. And so trying to take all of that and bring it together. I need a data scientist to do that. And funding, but yeah, it would be interesting to look at some of that, especially for things that are kind of, in the past. And we do see patterns sometimes for a particular compound, and I won't say what it is, but we had one that worked as an intramacrophage TB only. So you grew TB, that's a mycobacterium on its own in vitro, didn't touch it. But if it was inside the macrophage, it killed it. That same thing works against triple negative breast cancer grown in spheroids, but not grown in 2D. So it's what it's doing. We're not really sure yet, but yeah, it's interesting. But to get all that data out there would really actually take quite a bit of work. We don't have that as well organized in that data, all that data. I have a database, but it's not as well organized. I guess I'd get Steph on it.
Yeah, I think it's interesting too, since obviously this was decades of work, what the technology was like when we first started and what your first database was. And then, Stephanie Farrington's on the, out there in the audience, but she was also recognized for her contributions for the database. So it evolved, and John and Stephanie really changed it. So now when we do cruises, it's very different. We get a lot of data right away. It's almost real time. Do you want to talk about that at all, John? How it used to be that, we just wrote things down.
Yeah.
Kind of when we had time, we
I mean,
But now we do everything real time.
yeah, I mean, the submersible, as wonderful as it is and was, you know, two people went down, two scientists went down the sub, and you wrote notes in a notepad and took photos and collected samples. Now, since we lost the submersibles, we do all of our deep water collections using ROVs, and kind of like this Zoom chat where here the Zoom could be seen by hundreds and hundreds of people at the same time versus just having a seminar at Harbor Branch where a limited number of people could see it. With the submersible, you're seeing a live feed on the ship, what the ROV is seeing while the dive is going on. So for example, we had a cruise to Cuba a couple years ago, and it was truly amazing. We had 5 different computers set up in the lab, and each computer station, we had one station for the fish, one station for sponges, one station for algae, one for gorgonians and coral. And each team of scientists, both from the United States and Cuba, had a computer that logged in into this database in real time so you could identify stuff as this ROV was diving. And then that data would link up with the ROV data, which every 2 seconds, we have an exact position of the ROV and the depth. So all that, once I walk off the ship, all of that data is already compiled, exactly latitude, longitude, depth of every photograph, every sample, what it was identified as by specialists on the ship. And what NOAA Oceanus is doing is a step further, where they're using that live feed throughout the world. Anybody can log into it and watch the live dives as they go on in the NOAA Oceanus. Personally, I'd much rather be in the sub because you're in an aquarium. You go underwater and you are in an aquarium. And even better than that is scuba diving, because then you are in the water. So that's my favorite, scuba diving. Then sub, and then ROV.
And you got a lot more bottom time. And you can go a lot deeper. So
a question for you, Dennis.
Yeah.
Did you see that one? Did you want to, you mentioned the awe-inspiring deep algae. Were these Ocophyta, or was it a mosaic of red and brown algae?
The ones that I mentioned?
Yes.
The ones in the Bahamas, the deepest algae are very thin crustose red algae. They have very low metabolism, and they can apparently live at extremely low light levels. The ones in Pulley Ridge is big fleshy green, and it's kind of like a cabbage head, and that's the only place that it's been reported anywhere in the world. And again, it's kind of a long story, but yeah, there's a lot of diversity actually with the deep water algae. And you mentioned somebody, I think John mentioned Hawaii, and there's some really great work that's been going on with mesophotic algae from Hawaii. So I really would like to go there too, John. We ought to think about that. Shirley's been also answering some of these things that are coming in. I think between all of us, we've answered most things.
One more. No, I think there's one that hasn't been answered, and
okay.
There's one about the weirdest adaptation.
Oh yeah, I was gonna go there. I was gonna save that for now. So you want to do that? I was gonna go to you first, John. So the question was for each of us, what is the weirdest adaptation that we've seen, I guess, in our collections of various organisms? I don't know if dead baby doll parts count on this one or not.
No. I think this actually is, I would think, is the carnivorous sponge. I'll let Shirley talk about that.
Yeah, the carnivorous sponges, have been reported from really, really deep water. I remember the first time we found one, and I think it was off the, and this is another example of like this database now and how we have all of these images available. But I think the first time we found one was maybe off of the Keys, and I was so excited because this is a sponge that has become adapted to living in kind of like sponges are filter feeders. They filter particles out of the water, and in areas where there aren't a lot of nutrients, they've actually adapted to be able to capture things like little crustaceans with the spicules, and they can, with their spicules, which are little microscopic skeletal elements, and they can digest them. And the cool thing about our database now is that since I've been looking at these photos, it's like, oh my gosh, that's a carnivorous sponge. We didn't realize it was a carnivorous sponge. So we have many more of them in our collection than we thought we had, and from lots of different areas. So that's probably high up on my list of what I'd like to investigate in my spare time.
How about you, Amy?
What was the question? I'm reading other stuff.
Was there anything in particularly, in terms of weird adaptations of things that you've collected or that you've seen in your submersible days?
I don't know about weird, but there are certainly cute things like those. They're like crabs that carry around hexactinellid sponges and things, and they're super neat to see. But I don't know what the adaptation's for, why they carry them around, but they have their little hat on them, and I don't know, strange.
So one question that came in, I know Shirley's working on it, but John, I think this is kind of up your alley. Are you allowed to collect black coral these days, or is it against the law? So what's the regulations on collecting black coral?
Well, each country has their own regulations. Black coral is part of the CITES. So if we had a permit, if we went to another country and had a permit that allowed us to collect black coral, we would have to have documentation that we had permission to collect it from that country. And when we brought it into the United States, we have to show that it was CITES approved, it was allowed to be collected. But basically in the United States, you cannot collect coral of any type, hard coral or black coral, without getting a scientific approval through NOAA or NOAA Fisheries.
Okay, thanks. Well, I think unless something else comes up right now, that's pretty much our time. And we usually try to end right about 5:10. It's 5:10. So I think we should probably end it here. And I would just say thank you, Shirley and John and Amy. And I think the four of us really have talked about trying to do something again next year. We probably got something else we can come up with, another talk in us somewhere, or at least we can sit on the couch in the auditorium when everybody can come back live and we can tell some more tales from the sea. Shirley, you have anything else?
Now, again, I just want to thank each one of you as well. I want to thank, we still have 68 participants, so that's amazing. So thanks very much. And I think I'm speaking for all four of us when I say that this has just been a fun project. I mean, it's a lot of work. It's been a lot of, and it really has been a lot of work putting all of this together. But for us, it has kind of re-emphasized how valuable this collection is, not only for the drug discovery program, but for many other applications as well. So we're pretty excited. I mean, this is the beginning now. We've got the data there, it's accessible, and now we can start mining, we and others can start mining those data and apply them for research, resource management, education, and outreach. So thanks very much.
Amy, what about you? Got any final things you want to say?
No, thanks for coming.
John?
Yes, I'd like to give a shout out to Barbara Mills, who's my volunteer for many, many years. Hello, Barbara. And she and I started this whole project almost 18 years ago, when we first started digitizing all of the 35mm photographs. So Barbara, you were a great help, and this would not have happened without your help, and also Fred, or Jim Fredlock.
Oh, that seems like a really nice way to end things today. So thank you all. And we hope to see you back in 2 weeks.